Hello everyone, our presentation is nitrogen fixation contributes to nitrogen loadings of Utah Lake. It's presented by me, Han-Yan Lee from the University of Utah as a PhD student, and my professor is Dr. Ramesh Gual from the University of Utah, and our collaborator Dr. Throm Miller from Jordan River Farmington Bay Water Quality Council. Surface water quality has become a worldwide issue. According to USGS, toxic harmful algal blooms have become implicated in human and animal illness and death in at least 43 states in the United States. So some of the examples are from Great Lakes like Lake Erie, Lake St. and the well-known Utah Lake from the state of Utah. As for the effects of harmful algal blooms, it does not have the ecosystem effects like decreasing dissolved oxygen, which will cause less variation in aquatic habitat, death of aquatic life, and also it can pollute downstream water resource, and it can also produce some harmful toxins like cyanotoxins, which is harmful to aquatic habitat, mammals, and humans, and it can also cause mild to severe illness and sometimes even life-threatening illness. Cyanobacteria is different from the algae we usually know because cyanobacteria are a group of oxygenic photoautotrophic gram-negative bacteria, while algae are eukaryotes. So from the evolutionary tree, we can see that cyanobacteria appeared earlier on Earth from 3 million years ago, while algae appeared much later and is more close to plants. And also cyanobacteria, many are toxic, and some can fix atmospheric nitrogen gas into ammonia, and they are more resistant in the nutrient limiting conditions, while algae can cause nasty environment, but most are non-toxic, and they bloom in rich nutrient environments. Another question is what conditions can trigger harmful algal bloom? Phytoplankton is affected by excess nutrients in the environment and also affected by a certain temperature. Climate change with increase of temperature is usually no control over directly, so we want to control more about the nutrients part. And the question is usually for nitrogen and phosphorus, which one is a limiting nutrient? So if one is more abundant, another one is more limited. Conventionally, there is more phosphorus limited environment, and nowadays with more nitrogen input, and we really believe there are more nitrogen and co-limited conditions. So however, where is the source of nutrient come from? Whether it's only from the nutrient loading and it can also from the internal cycling of the lake is also questionable. So nitrogen fixation is an important component of nitrogen cycles in the ecosystem. We can see from both of oligotrophic and the eutrophic lakes, nitrogen fixation and phosphorus recycling from sediment are both very important components and pathways to add nutrients to the ecosystem. And more nitrogen fixation and phosphorus recycling occurred in eutrophic lakes to support eutrophication and also because of initial abundance of nutrients. From the right cycle, we can see that nitrogen gas can be fixed to ammonia under anaerobic conditions, and this form can be converted into other different forms of nitrogen compounds with the support of oxygen. And these forms of compounds can be consumed by algae. Nitrogen fixation can play significant roles in adding nitrogen to the ecosystem. It's saying that biological nitrogen fixation supported by prokaryotes appears important in making up deficient in nitrogen availability, contribute to the phosphorus limiting status. And also, the planktonic nitrogen fixation tends to be low in oligotrophic and mesotrophic lakes, but it is often high in eutrophic lakes. And also rich of the fixation in cyanobacteria mats could be high to very high, but mostly occurs and covers only a small area of the ecosystem. And some of the water bodies could have high to like higher than 20% of the nitrogen input by nitrogen fixation, and even high to 80%, 86%. Biological nitrogen fixation in bacteria is conducted by special cells called heterocyst. So it's mostly in filamentous cyanobacteria, and compared with the common cells, it has a special shape, and the nutrients and other information compounds can be transported from the common cells and the heterocyst. The common cells is called a vegetative cell, and it can carry out photosynthesis and can differentiate into heterocyst when the environment has a lack of ammonia and nitrate, while for the heterocyst, they carry nitrogen fixation from atmosphere, and they're surrounded by thick cell wall to prevent oxygen fatal to nitrogenase, and the entire process energy is provided by light. So this function usually predominate nitrogen limitation environment, and there is usually no method to resolve the nitrogen fixation issues when the species is here, unless we can manipulate nitrogen and phosphorus ratios. So nitrogenase is the enzyme reducing atmospheric nitrogen to ammonia, and is responsible and catalyze this process. It is sensitive to oxygen and requires oxygen and anoxic environment, and this protein is affected by temperature and pH. So this equation shows nitrogen and with 8 hydrogen ion, and can be converted to ammonia compound with the help with the nitrogenase. This enzyme is also coded by genes called NIF genes, and these genes can be quantified in the bacterial cells. Another question is how bacterial genes can finally become the protein or enzyme. This process is called central dogma, which DNA can replicate themselves and transcripted to RNA molecules and then translated to the protein. So imagine we have a cluster of functional genes in the cells with double strand, and the first step is it can be transcribed to message RNA, which has all the information of this gene, and then with the translation, this RNA can guide the formation of amino acid, and with the folding of amino acids and peptides, it can form proteins and enzymes and other functions that the bacterial cell need. As nitrogen fixation is very important, how can we identify this? One way is to use acetylene reduction assay. It uses the ability of nitrogenase to reduce acetylene to ethylene. In the acetylene reduction assay, um, it converts acetylene and the two of the hydrogen compound to ethylene, while in the biological nitrogen fixation, it requires 8 hydrogen compound. So for this one, it's actually the rate would be 4 times higher than the actual biological nitrogen fixation. So the theoretical conversion factor is 4 to 1. There are some advantages and disadvantages of that. The good thing is it's really low cost and sensitive. However, both of the compounds are gas format and need to analyze by gas chromatography, and it really needs incubation time from 15 minutes to 4 hours. During this time, people usually use serum bottle and which will have oxygen accumulation and also depletion of the carbon dioxide, which all may affect this process. The procedure of acetylene reduction assay is first you prepare 160 ml of the serum bottle and which has a aluminum cap and a rubber septa. And with that you add 100 ml of the water and leave around 50 ml of the headspace. And then you inject the acetylene gas from the acetylene tank, which try to make the headspace of 10% in the atmosphere. After incubation for 15 to 4 hours, again you take the gas, which right now have the acetylene, air, and ethylene, and 0.5 to 1 ml of the gas can be injected with a syringe into the gas chromatography with a FID detector, and then it can be analyzed by the GC, and there will be 2 peaks shown in the system. One is the acetylene and one is the ethylene production. According to the amount of ethylene production, we can quantify the biological nitrogen fixation rate by the converting factor of 4 to 1. And another method to identify nitrogen fixation is to use molecular techniques, which can help to detect gene copies and also gene expression. So the gene identification is to detect whether the nitrogen fixation genes and species are actually present in the environment, and the gene expression means whether in such conditions this nitrogen fixation function is working or not. So if we consider gene identification is whether the species or certain gene is present or not present, then gene expression is to identify whether it's working or not working. So gene identification is based on the quantification of DNA molecules, and even if we detect the presence of species, we can't see whether the function is activated or working. But however, with the detection of the RNA molecules, we can sure that certain function is working and it's transcribed to protein right now. But the thing is, RNA is more fragile, and the samples really need to be preserved as soon as possible. And the detection of new functional genes and the gene expressions are similar to any other genes. So the first thing is we want to know which gene we want to target. And from here we know like what primers to use and about the assay design. And then after that we can make sure our compounds can be amplified in a certain machine. And these compounds are bound with some fluorescent signals, and with amplification the signals become significant and significant, and it can be released and identified by the instrument. And then through the software analyze, we can get to know what's the initial copies of the gene and gene expression. So there is a case study we conducted on Utah Lake for nitrogen fixation. In 2018, we conducted sampling monthly on 7 sites across the eastern part of the lake. We conducted gene quantification and gene expression of the NIF gene, which the primers and experimental design were conducted in an EPA lab. So we found that phenizominin and dolichospermum were two potential nitrogen fixers, and their presence were positive correlated with the NIF gene and gene expression, and their dominance was found in August across the entire lake. So this study was published in Water Research this year. So some results we found that is for the open water area, the orange one is a phenizominin, and the gray one is a dolichospermum, and their presence and relative abundance increased from June, and the highest was found in August. But in Provo Bay, it has an early bloom of dolichospermum, and the maximum one was found in June. And also, when we quantify the NIF gene, we can find that in the open water, the gene copies are increasing continuously, while for the Provo Bay area, the highest one was around June and late June. So similar results we also find for the gene expressions, which means probably the Provo Bay is still the origin of the bloom for that year. And similarly, this year we also conducted some sampling with the help with Dr. Thoron Miller's group. We sampled eastern site 5 sites, and this year we did not only do gene quantification, but we also did a acetylene reduction to analyze and measure the nitrogen fixation ratios. So it was found that high nitrogen fixation rates we measured once in August, and it with the presence of filamentous cyanobacteria. And the NIF gene and gene expression were also quantified and to confirm this activity. So when we calculate the nitrogen fixation rates, we find that the highest nitrogen fixation rates were measured at the mouth of Provo Bay. And the highest nitrogen fixation rates we measured in situ was 0 to 0.73 nanomole nitrogen per liter per hour. And when we come back to the lab and measure using the best condition and saturated light, we measured higher rates, and it could be as high as 2.52. So when we use this for calculation, it results in a annual loading of 0.31 gram per square meter per year, and it could be up to the mesotrophic or eutrophic level of the lake during the bloom. And when we measure the gene and gene impression results, there were positive correlations between the nitrogen fixation rates. So when you see the right figure, the second column is the mouth of Provo Bay. It has the highest nitrogen fixation rates and also the gene expression and gene quantification. However, when you see the Geneva discharge of the fourth column, it also has the highest nitrogen fixation rates. However, the gene was lower. So when we consider the results, we should combine both of the methods to see. So for the mass balance of the lake, the input of nutrients can be from varieties of sources, and except for the non-point source we usually know about the wastewater treatment plant, there are actually many undefined sources. They may also play significant roles in eutrophication. For example, there's a stormwater input, agricultural runoff, and also atmospheric deposition. So another way for inputting and outputting could be the nitrogen fixation, and we measure for the Utah Lake. The estimated could be around 11,000 kilogram nitrogen per year. Just consider 3 months bloom period, and for the nitrogen reduction, it could convert nitrogen compound to the nitrogen gas and release back to the atmosphere. So we measure nitrate reduction. It's not just only the denitrification as a soil compound. It also absorbs some nutrients into soil and can be released to the water column at some time. So for the future improvement of our study, more sampling and data points can be conducted. Of course, it will give better estimation of the total nitrogen fixation. And for the lake sample with low nitrogen fixation rates and low concentrations of cyanobacteria, the sample may be concentrated to be estimated. And also in the lake water, the concentrations of other nitrogen compounds may be measured to estimate the amount of nitrogen fixation contribution to nitrogen cycle. So we want to thank the audience for attending this presentation, and also thanks for the technical support by EPA and Utah DWQ. And also we thank the support by Dr. Thoreau Miller and his group for the contribution to this study.